All right. It's 11 o'clock. Good morning. Good morning, everyone, and thank you for attending the second webinar, the Netpore webinar of the first session. I'm very, very delighted today to introduce our two speakers of the day. We have managed to maintain this format of having one speaker from the academic world, who is Elder Santos from the University of Groningen. And we also have another speaker, who is Gerard Macias Sotuela, who is from the private field. And so we will try as much as we can to maintain this format, to have the vision and inputs in this field of porous material from the two worlds, and make sure that we maintain this connection. So we will start with Elder today from the University of Groningen, and he will talk about porous material for solving medical problems. He will let you know more about that. And Elder, if you would like to share your screen.
Yes, thank you.
Happy to stop.
Yeah,
sharing.
exactly.
Okay.
Thank you very much. So thank you very much, Frederic, for the kind introduction and also for the kind invitation to be here. It's a really great pleasure to talk to you all today. I believe you are seeing my screen?
Yes.
Full screen now, right? Yeah? Correct?
Yes, we can see your full screen.
Great, thank you. Thank you very much. So today I will tell you a little bit of what we do and what we have been doing on this field on porous biomaterials in very, very general, just to advertise a little bit our expertise and our tools that we have in-house. Yes, we do use this kind of porous materials more for biomedical applications. So we are not so much on the fabrication side, but we are much more on the application side. So I will tell you a little bit of everything that we can do with that. So as Frederic mentioned, I am at the moment University Medical Center in Groningen. So I'm the head of the Department of Biomedical Engineering. So here is the group, but I still have my group in Helsinki as well. So here we are more focused on biomedical applications, and in Helsinki is more on pharmaceutical applications. So 2 labs in 2 different countries. And here you see a lot of different kind of funds that we have, projects. I'm also coordinator of a big Marie Curie ITN network, P4FIT. This is for tendon regeneration. In addition to Netpore, I also belong to the Nano2 clinic cost action that is in place at the moment as well. We're in Netherlands, so you probably know that Netherlands is a very small country here. And we are very up north, so from Amsterdam to Groningen is about 2 hours train. It's a very beautiful city, and our university has about 35,000 students. And here you can see the front door of the UMCG, where we have about 12,000 employees, and is very clinical oriented. And you see here it's pretty much in the middle of the city of Groningen, and most of our research is clinical oriented. So we are working very close with doctors, medical doctors, but of course we are in the clinical side. So the University of Groningen is quite old. A few years ago, we celebrated 400 years. Even though they are two independent institutions, they make one. So University Medical Center and the university itself, they kind of merge together. And in the case of UMCG, so is really patient oriented, care oriented, but also education oriented for medical doctors. And we have different kind of institutes. So you see here some of the institutes that we have: cancer, brain, the drug, and the material science engineer. That's where my lab belongs. That's where my department actually belong, and also the health aging. So we have the European Center for aging, biology and aging. More in concrete, let me just I take opportunity to introduce my own department. So as I mentioned, we try to solve medical problems, and nothing better than work close with the clinicians, because they really know what are the medical needs. And these are some of these are the 8 groups that we have currently in my department. So we do work a lot with biomaterial interactions, so biofilms, bioadhesion, biotrobiology. So we can actually test the different interactions between the materials and cells, for example, bacteria. So material interactions is one of expertise of my department. Nanobiomedicine, imaging and analysis. So we also work a lot with physics and optics. Nanobiomaterials, surgical robotics. So we also develop small robots, magnet-based robots that can actually do some job when they are implanted inside of the body. And then my lab here, it's more focused on tissue regeneration, and I will show you some examples later on. So this is actually our group or our department here. We are about 70 people. Most 50 of these people are actually PhD students. And this was actually taken just 2 days ago, this picture. But let me introduce you our hospital. So I hope you were able to follow the video. So this is our institution, and you saw a lot of core units and a lot of instrumentation that we have. So we have all the methodology, all the techniques to analyze surface materials. We have also animal facilities, so we can go from zebrafish to small animals up to rabbits. And obviously, we have just in about 200 meters the patients that we can also interact with. Okay, but let me tell you a little bit of what we do specifically in my group. We started in Helsinki, and now we are continuing here in Netherlands at UMCG. So this is pretty much what we do. We work a lot with nanoparticles, and lately we've been a lot interested with hybrid nanomaterials. And we do a little bit of everything for drug liver applications, for tissue and implants, and then we also work with trigger materials for photothermal therapy or photodynamic therapy. We love this kind of materials that we can chemically modify in order to achieve some results. So let's say in the past years, what we have been trying to do is to take different materials, and porous materials are just one of the examples, but lately we also have much more of those examples, and we try to give them an application, and of course always in a biomedical application. And you see here some of our beautiful covers that over the years we tried to apply for different biomedical applications. Cancer, cardiovascular disease, and diabetes are something that we have been really very keen in trying to solve or in trying to apply these materials for this kind of medical problems. Okay, so this is actually the summary. So we have been working a lot on target cancer immunotherapy, diabetes, heart disease. Most of these ones have been with porous silicon materials. And then we have also worked with other kind of materials that actually they are also can be made also porous, like lignin structures, or like scaffolds, porous structure scaffolds for tissue regeneration. So we have been trying to modulate and to find a combination of different kind of applications. So together with actually Jarno Salonen, so from Turku in Finland, we have had a long history of he actually fabricated a lot of these materials, and then we applied these materials for biomedical applications. Jarno is going to talk at some point in these webinars as well. So this is just an example. We got the particles from Jarno, then we load them with different drug molecules, and then we modify them in my lab for cancer applications or others. And we went down to the, well, not to the, here it shows the human, but we never tested in human, but we tested in vivo in small animals. And this is just different kind of applications that we have done with porous silicon materials themselves. So bioimaging, biodegradation, we even made pills out of this because I was in the pharmaceutical faculty. So we did pills of this. So this is exactly the same kind of pills that you can find in the pharmacies. Targeting to cancer. We have also been very much interested now on immunotherapeutical applications of these materials, viruses as well. I will show you some examples. And lately, we have been very keen on trying to formulate these structures, or combination of these structures, by using techniques, new techniques, the microfluidics technique that I will tell you a little bit later. So when it comes to porous, my lab has already produced a lot of different PhD theses. So these are all the students that throughout the years have worked one way or the other on porous silicon and silica materials. And these are two of the books that with many of you actually here in this Netpore, we have written, or we have assembled together, and I've been very happy to be the editor of this book. So this one here, it was just last year, and the first one was in 2014, and it's basically based on porous silicon materials. So as I mentioned, we are not so much involved on the first part, that is the fabrication, but we are much more involved on the later part where we already have the materials and then we just modify these materials and apply it. So this is an example of porosilicon solutions, but here you see also silica-based porous structures, or this is porosilicon, and this one here, which is a diatoms. So we also been working a lot on diatoms, silicon materials in the past. So this is also a kind of porous structures that we like to work with. With porosilicon, we have been doing a lot of chemistry in those, especially combining different kind of structures. So in the past, in order also to stabilize the porous structures after we inject them, so we have been assemble or we've been combining with different kind of polymers, hyaluronic acid, for example, bilayer of polymers on top of these structures. Nanocomplex, so we combine porosilicon with porous structures, and then, for example, gold nanoparticles for different kind of intercellular activations, and even with the DNA capping porosilicon structures in order to have an intercellular response. Needless to say that after this, our lab is very expert on looking into detail into the intracellular interactions or drug delivery and with the intercellular phenomena. So how these materials reach the cells, how they interact with the cells, and now what happens once they go inside of the cells. So this is just one example where we have been doing. We have been working on diabetes, where we do oral formulation of porosilicon materials. We modify these structures of this porosilicon. You see here with albumin loaded with insulin. We have a polymer around these particles to stabilize it. We have also modified this with FC conjugated structures in order to actually target them to the intestinal wall. And insulin is an example. GLP is another example, another peptide. And then we follow, as I mentioned, we characterize all these structures. So TM, DLS, and so on. So the typical techniques that we have available in our lab. But more than that, we have then models that cellular models that we can test. So here you see an example of intestinal cell layer that we can evaluate the interactions between these nanostructures and then the intestinal wall. So we do this with in vitro, or then we do this also ex vivo. So we also collect the intestinal wall from rats, and then you can evaluate exactly the same thing. So we can do in both ways. Of course, we try to do as much as possible on in vitro models, 3D in vitro models as well. We all know that injectables are always problematic. So in the past, or what we are also very good at, is to test the interactions of the surface chemistry, different surface chemistry of porous materials. Again, I show you here the porosilicon and what effect they have, or what kind of interactions they have with proteins, for example. So and this is human blood proteins, of course. So we can have access to human blood, to patients as well, and we can make these mappings where we very precisely detect or are able to evaluate what kind of proteins interact the most with different kind of surface chemistries, and what effect that has then on the biofate of these materials once they are injected. But we, as I mentioned, we have also worked with other polymeric-based structures that they are also porous. This is just some examples that we did very recently. And in this case, the applications are totally different. They are polymeric-based structures. In this case, we use it for spinal cord injury. Or then we can also combine them with biological structures like exosomes. So we just combine inorganic structures, porous inorganic structures, with these exosomes. I will show you some examples so that we can use it, well, in this case for cancer, but I will get back to that. So porosilicon is very nice, or it was very nice for us because the chemistry was very simple, was very easy, and we also could modify the surface of the particles with imaging radio tracers or even with fluorescent labels. So we have done a lot of this in order to follow them after injection. So this you can see, and we have used SPECT-CT, so these are positron emission tomography imaging techniques that they are also used in the clinic, and you can see where the particles going, where they accumulating, and you can also evaluate if your material is stable or not stable, if it reached the tissue or not. So we have all these tools available here as well in our hospital, where you can follow very nicely where they are accumulating over time. So this is for small, these are rats, but we can do it up to rabbits, as I mentioned. So lately, we also been very much interested on working what we call transformable drug delivery systems. What does it mean is that we start with a very large, with some structures, and we somehow modify the structure. And this can be in terms of the size, it can be in terms of the shape, or it can be in terms of the formation of these materials. And typically, we combine inorganic materials with polymerical structures, for example. And I've been giving you the example of the cancer, but it's not only valid for the cancer. So this is an example that we have then also for heart tissue. So this material, the inorganic porosilicon materials, for example, we modify and we follow how do they reach actually the infected myocardium, and you can use exactly the same kind of tool. So you can surface modify these structures with, for example, peptides. So we have been also very much interested in the surface modification of materials, inorganic materials. With some ligands and biological moieties, for example, peptides, small short peptides that are targetable to different tissues and organs. Well, in this case, it was to the heart, as you can see in this image here, in this SPECT-CT image, you can follow. But we have been building up, as I mentioned, these transformable materials. Here I show you just a very brief example where we modify porosilicon with aptamers. So we build up different polymer structures and aptamers in the end in order to target them to cancer cells here. So you see here in vitro some cancer cells. So this is one way how to do it. Or then you can use light to trigger to go from bigger structures to small structures. So we also have been doing quite a lot on that. Oh, oops, sorry. So here you see, so again, in porosilicon structures, and we build up these photoresponsive structures on top of porosilicon. And in the presence of the UV light, we have a response. So we go from a very stable structure to a much reduced structure that can penetrate then the cancer cells in this case. And we are able, of course, to modify the structure to follow what happens to these materials once they are injected. So here again, we look into the protein absorption and if they are stable and so on. So we are very much interested to test all these kind of materials for different kind of applications, particularly if we are able to modify the surface of these materials. So this, I'm not going to go into detail, but just to let you know that these are all the biological tests that we can do, from cytotoxicity to the uptake, and basically with any kind of cell that you can imagine. So recently we also been trying to combine a little bit the inorganic materials with structures that can communicate with the cells, and of course they are also light responsive. So what we call nanoparticle cell communication enhancement. So again, we use porosilicon material here. We modify them with this beautiful peptide structures that they have different functions, that we did a lot of computational analysis in order to see how do they behave in terms of the organization on top of these structures. And then we actually were interested to see what their response was when in contact with different cells. For example, with macrophage cells, we didn't want them to actually interact too much, but with the cancer cells, we were very much interested to get them inside. So we did a lot of in vivo, of course, so you can see here a lot of you can visualize where these particles go. So if you look into your right side, so you see accumulation of specific accumulation over the time of these materials, and we are able to not only visualize where do they accumulate it, but also to quantify then how much it has accumulated inside of these tumors. And of course, because we are we have been in the medical universities, we are also able, of course, to look directly into the tumor, so we can extract all of this. So my group has a lot of experience on this, so we can evaluate single tumors as well and tumor volume response. And to finalize, I just want to show you also a little bit of what we've been developing for the past few years. So what we call biohybrid nanovaccines. So we combine structures. It can be here. I show you two examples: viral, but also porosilicon structures, where we combine with the cancer cell membrane. And we wrap them around. So we make these constructs or composites in order to induce immunotherapy for immunotherapeutic applications or immunostimulation, let's say. Let me just move on. I don't want to show this. So what we've been doing is we've been using for melanoma and with combination also with clinical applications. But here you see very easily. So this is a porosilicon structure. We wrap around with a polymer, and then we put all this into a cancer cell membrane, and then we evaluate the effect this in terms of immunotherapeutic applications, and we demonstrate that actually in melanoma it works very nicely, and it works even better when we combine this our nanovaccine, as I call it, with the clinical application. So in this case, it's a drug checkpoint inhibitor actually. So you can actually have an effect of this, enhance an effect for cancer therapeutic applications as well, but you can do it even more difficult, or at least for me, I thought it was very difficult, is that you can feed cells with inorganic particles, porosilicon particles again. You can feed the cells, collect the particles on the other side, and expect that they were somehow surrounded by these extra vesicles, solar vesicles, and then inject. So these are cancer stem cells, and then inject them back, and then you hope that they would actually find the cancer by themselves without any targeting moiety on the surface. And we actually were very surprised to see that this was actually the case. So when we look into the tumor growth, we saw that this kind of approach had a much strong effect than, for example, the anti-cancer drug that is used in the clinical, and the survival rate was actually much improved as compared to the clinical applications. So this was another way how you can do it. You feed also the cells, you collect the particles, and then you give them back in order to increase the response, tumor anti-cancer response. And this is my last example, I believe. So we also combine these cell structures with the porosilicon structures with light. So we love also a lot of light. So in this case, what we did is that we created porosilicon. We actually had gold nanoparticles on top of this sputter, on top of the porosilicon particles, and then we wrap this construct around with cancer cell membrane, and then we study the heating and the light response. And when we gave that to the animals, so as a vaccine again, so just to show you, so we are very able to do all these tumor models as well in our lab. We saw that, well, you see here the tumor growth compared to the controls is much better in our case with this porosilicon, and you see also here that the survival rate is much more than all the controls used. So this is a way how we can use porosilicon materials to lead to some of these applications. And oh, sorry, this was not my last slide. This is my last slide. So we can use also microfluidic techniques. I don't know if you are familiar with those, but microfluidic techniques are you can make nanoprecipitation, or you can construct these nanostructures or microstructures using these small channels And this is, for example, how some of the vaccines were produced in the COVID. And we also use this to make combinations of materials. So this is just an example what we did. Porous silicon, we inject in these channels, and then we cover this porous silicon with polymer structures, for example, to stabilize. And in this case, you can load several drug molecules. In this, we try, for example, with 3. So you can have 3 different drug molecules in one single particle, which is really amazing, for example. And then again, of course, you see all the in vivo and so on. But yes, so this is just to wrap up. So we take these materials, we like to modify the surface of these materials with all kind of biologicals that you can imagine. Nowadays, we are also working in the combination of porous silicon materials with RNAs, special siRNAs. And then we do all the biological testing, since the injectables, oral formulations, topical formulations, and then evaluation, what happens, toxicity, uptake, drug release, and so on. So very fast, but that's what I wanted to say to all of you. Thank you for your attention. I'm happy to answer any questions if there is time.
Thank you very much, Helder. It's for this great presentation. It's a beautiful overview of your activity, and it's very impressive. I would say.
Thank you very much.
And very, very interesting. You would need 4 hours.
Yeah, almost, yes.
Yeah, it's beautiful. So if there are any questions, please just go on.
And my contacts, of course, are in the beginning. So.
Hi, good morning. I am Luis.
Hello, Luis.
Hi. First of all, I thank you for your very nice and impressive presentation and the
thank
facilities
you, Luis.
And all the things you can do in your labs. This is amazing, and a lot of possibilities. So you are doing a great work. So my question is something, just a curiosity about the particles, right? So you are using one type of particles. My question is, what is the role of the geometry or the size of the particles? Because they are one part related to the material, another part, important part related to the functionalization, the chemistry of the surface. And it's very important, so the geometry of the size and also the degradation of the particles. So
yes,
in some cases you want, some stable particles, and sometimes you want some degradation of the particles because you want to remove the particles. So can you comment a little bit about this point?
Thank you very much, Luis. Yes, of course, you touched very, very important points, which are very, very important for the biomedical application. So in terms of our particles, we always use, in most of the cases, thermal hydrocarbonized or thermal oxidized porosilicon materials. This is basically the whole history of this what I presented. We always work with spherical particles, or well, as much as spherical as we can get from porosilicon materials. But we have not tried any kind of other kind of shapes. Typically they are mostly round or round-like structures. And the sizes, it depends. So we have worked from 150 up to 300 and so, I think never above 400. And yes, you are right, depending on application, it's very important, not so much the geometry, but the size. It has a crucial importance for the biomedical application. So we have seen that for targeting purpose, of course, the smaller you want for injectables, between around 200 is the ideal. So this is what I would recommend. But more importantly is what you put on the surface. So the porous materials themselves are not so stable, and therefore you need to modify them very properly in order to get them to the right place, for example. And of course, after you touch another point, that is the degradation. So that's very important. Our porosities are typically around 60 or 70. So they will degrade faster or slower, depending. But we do want them to degrade. So of course, you don't want to have this over the time. So, but we don't care so much if they take several months or several weeks to degrade, as long as they degrade and the degradation products are not toxic. And this is what we also do test. We are not so able to test them in vivo, but in vitro we have done a lot of testing on this degradation, the biodegradation products, and their effects to this. So, for example, the production of radicals to the cells, because we know that the degradation products some can induce some inflammation and some stress to cells or to organ or tissues. So we have all evaluate that, and it's very, very important when the medical application. Doctors are very concerned about all this, indeed.
Thank you very much, Herder. Very nice presentation. Thank you.
Thank you. Thank you.
Other questions?
I do have one for Helder.
Yeah.
So I'm Gerard.
Hi.
I'll be presenting afterwards.
Yes.
And I have this question about why are these particles still in research and not in commercial development to making drugs? Because it has been several years where different designs of particles with different approaches have shown that there is a good decrease in the size of tumors, for example, in cancer. However, even though a lot of years have passed, I don't see any applications of that in the pharmaceutical industry as of yet. What do you think is the reason for that?
Yes, very good question, Gerard, and you touch now the wound of the field here. Yes, I mean, I've been wondering myself, because we have a lot of data, we have a lot of demonstration that these particles would have a great future. I mean, my feeling is that is a question of investment, is a question of getting really a company that takes this till the very end, and we have. I think the field is missing that kind of approach, that or that kind of pharmaceutical company that is really interested to go there. And before we get FDA approval of this kind of materials, most likely the companies will not be so much interested, for example. So it takes. I will put it in a very direct. It takes a kind of a COVID situation in order to realize that we need to apply this material somehow. So we would need this kind of shaking into the pharmaceutical companies to really see the value of these materials, to take it to the next step. So I guess it's a question of investment, is a question of to getting the attraction of a strong company that would really be interested and keen on these materials. And we have discussed a lot with the different pharmaceutical companies, but they have never been so attracted, because they have polymeric particles, they have liposome particles, and they always want to play safe. And this is the big problem, and it's also a very high competition for the porous silicon materials, for example. But I still believe that one day we will get it.
Thanks.
Yeah, thank you.
Okay, thanks a lot, Helder. Thank you very much for this great presentation.
Thank you so much.
You're welcome. Thank you. We will move to the next talk from Gerard. We'll just ask a question. If you want to share screen, Gerard.
Yes, of course. 1 second.
So Gerard is actually working in a company, which is his company. And so we will have a different point of view on nanotechnologies. This company is Masia Sensors. And I apologize for the typo on the web and even in the email. It is not located in Spain, but it's in Scotland, what you said. And I'm sorry, because it wasn't corrected. Fine. So Gerard,
no problem. I understand the confusion. After all, our website is in Spanish. So it's an understandable error.
Okay.
Well,
So
thank you so much, Frederick, for the introduction. I'm really, really glad that I have this opportunity to present Matthias Sensors and also present what we are doing to contribute to the rapid development of complex biosensing devices by what we call the lab on PCBs approach. And during this presentation, I will explain also why we are here in the Netpore cost action and how we envision that porous materials can actually be implemented into these lab on PCB devices. So let me start by just briefly introducing the company, Matthias Sensors. And what we like to call ourselves is biosensor developers. The reason for that is because if you want to take an idea from the lab to detect something biological and put it into an actual commercial application, there is no one single area of expertise that you need. You need several ones. And us are experienced in electrochemistry, surface chemistry, and optics. But also, in order to be able to provide all the rest of the expertise that is required to bring an idea to market, we partnered with a bigger group called the Zimmer & Pico group. And together, we can provide up to 37 different specialties that go from biosensing assay development to software, to reader design, manufacture, and also the fabrication of the final cartridges via an ISO 13485 certified facility, which is what we would need to be able to commercialize a medical device. So now that we have briefly presented who we are and what we do and what we are experts on, I would like to just briefly touch on what the lab on a chip device actually is. And we can see an example here on the right. And you can see there are lots of different channels, etc. Some wells, some detection device. And in short, the lab on a chip device is a way to integrate in a miniaturized system very common laboratory procedures so that they are automatically performed. And some examples are extraction of samples, preparation, reactions, as well as analysis. And one thing that I would like to say is that not all miniaturized devices that are a lab on a chip have all of these procedures. Some of them are simply for sample extraction and mixing, and then the analysis is done elsewhere. But in general, the overall trend is that you would do everything on these chips, from taking the sample to getting a result displayed. So if we look at the current state of the art of LOCs, lab on a chip devices, we can see that there are 8 different components that are mainly grouped into these 8 categories. And the first 5, the injector, preparator, transporter, mixer, reactor, and separator, are all microfluidic parts. Things like channels, mixing systems, filters, for example, for the separation, wells for the reactor, etc. And the last 3 are what most of us have mainly worked on, which are the detector and how to actually detect the signal from the biosensing assay. So the detector would be the biosensor itself. The controller would be the reader, and in a lab, it's normally a benchtop device. And the power supply would be something as simple as a cable from the controller to the mains in the wall. Or also it can be a battery for a handheld device like the one in glucose meters. Once you have been working with LOC devices in a commercial setting, you realize that there are some common issues. And there are mainly 2 issues that I would focus on mainly. And the first one comes with the microfluidic parts themselves. And the reason for that being an issue is that they are great, they work really good, but when you try to go to full-scale manufacture, normally the only way forward is to use injection molding, which is a very costly way of manufacture, unless you go to very, very high volumes, meaning you start selling all over the world and you actually have people buying your product. What this means is that mostly if you are trying to develop an LOC device, if you don't have your design really well made from the start, you end up using a lot of money on these injection molding parts, which can effectively lead to the company that is developing this device to run out of money and never get to market. And the second issue is the integration of the different parts of these components. There is a very common incompatibilities between materials. So an example, microfluidic parts are normally made of plastics. Some of the detectors are on ceramic substrates. Just having a good way of adhering these two is a common issue that arises, that of course lengthens all the design and development of the LOCs. And also, because of this amount of plastic parts that are normally used, it's very difficult to integrate other sensors, such as a temperature sensor that you could easily buy off the shelf. As a result, all of this makes an increased cost of LOC devices and a very slow design turnaround, essentially making all this development expensive and slow. So we analyzed what's going on here, and we looked for a different type of approach. So we turned to printed circuit boards, and we're going to explain why in a minute. And the reason for circuit boards is that it's a very nice system. It allows a way for effectively connecting multiple devices within a very reduced space. And thanks to that, you can have a small weight and volume for all the devices. Sorry, I'm just going to wait for a second. Pause there. We're seeing lots of chats. Okay, sorry about that. I was just wanting to check if there was any connection issue during the presentation. So going back to the printed circuit board. So printed circuit boards allow to reduce the weight and volume of electronic devices, which means that you can have a very, very complex system in a very small space. And proof of that is that you can see it everywhere. You've got them in your mobile phone, in the computer, in household appliances. They are absolutely everywhere. So looking further into PCB manufacturing, we discovered that they are really, really good for transitioning and doing lab-on-a-chip devices, or in this case, lab-on printed circuit boards devices. And there are 5 reasons for that. The first one is that their manufacture is scalable, and that's proven. You can see everywhere that PCBs are integrated into all electronic devices. So that's a check. The second one is that they are very fast to prototype. Overall, lead times to getting a new design to an initial prototype are anywhere between 3 days to 2 weeks, which is very, very fast and allows to test very different designs very quickly. Also, they are cost effective already from the R&D phase, and when you move to commercialization, the cost decreases even further, which is great for making lab-on-chip devices as well. As you can see from all the electronics, they are already designed to be able to integrate third-party electronics. So if you want to put a temperature sensor, picking up the same example as before, you can simply solder it on. And finally, there is this last reason that we were really keen on having, and is sustainability, at least a little bit of sustainability. And the thing is that with PCBs, you can readily recover the metals, which is a very nice thing to have, because most of the metals that are used in electrochemistry are very pricey metals, and there are normally conflicts all over the world because of that. So we decided to choose PCB as a way of making sensing systems. And to do that, we have an approach that has 4 steps. In the first step, we as a company, what we do is we develop modules. And these modules are different components that our customers will be able to choose. Essentially pick and place what they want. And with that, we move on to the second step, and we make the custom lab on PCB card. And once we have that designed and made, we are able to design a microfluidic system to move the sample around. And we finally assemble everything together into a cartridge that can be readily used. But of course, when we started doing some tests with PCB devices, we saw that there is one main challenge. And that is, how do we avoid the electrode corrosion so that we can actually make measurements? And the thing is that if you buy an off-the-shelf printed circuit board, they are not really going to work for making electrochemical measurements, which is one of our specialties, and where most of the biosensing devices out there want to go to, because of the simple way of measurement, of measuring the results. So that's where we started our product development. So we started first of all developing a metal deposition approach for electrochemistry that would not have this corrosion behavior, and that's what we did. Here we can see an example of how our process compares to the standard of printed circuit boards that you can buy commercially from anywhere. And you can see that on the blue line, which is the commercial available process from elsewhere, you can see what would be a corrosion behavior. And if you looked at the PCB in real time, what you would see is that you no longer have a gold surface. You would have a blackened device because it has been corroded. And thanks to that, we were able to launch in December last year our PCB electrode platform, which is a comparable system to the current screen printed electrodes, but made on printed circuit boards with our proprietary coating. And we have them available in two formats, one with all 3 electrodes with gold, and another format where we have a reference electrode made of silver. So moving on, we said, what can we do next? And we saw that not all systems commercially are good to be put into a flat device like a lab on a chip device. There's also a lot of implantable devices that are used for continuous monitoring. So we launched very recently a service to deposit metals for electrochemistry, that is normally gold, platinum, or silver, onto 3-dimensional electrodes. And what this does, it allows to have a very economic metal to provide mechanical stability and locate only the electrochemical metal on the surface, which is where you need it. And thanks to that, overall, it allows to reduce costs at the beginning, but also reduce costs much more at the end when you go into full-scale manufacture, because you no longer have a bulk of metal that is very expensive. You only have it where you need it. And that's where we are just now. But we still have some products in the pipeline that are already starting to be developed. The first one is an electrochemistry online course, because we found out that there is a need for good electrochemistry courses to learn the basics and how to make good measurements overall. But we are currently developing and preparing We are also getting our facilities ready to be able to do custom printed circuit board electrodes, where we are going to take the requirements from our customers, develop custom PCB electrode systems. And also we are working on getting surface chemistry coating services so that we can provide PCB electrodes with also a surface chemistry that allows us to readily implement some biosensing elements, such as an antibody, an antigen, a DNA, etc. And once we have all of this, we are planning on making a demonstrator system so that we can further reinforce the fact that we are able to do everything from the development to a full device system. And for that, we are going to focus on biochemical affinity testing systems. So these are systems that allow you to measure the strength between two molecules, such as an antibody and an antigen. And for example, that's used a lot in biologics research in pharmaceutical companies to find out which one of the drugs that they are developing is the best for their target. So that's everything about our company, what we do, why we do it, and what we are planning to do next. And now we just wanted to touch up on how we see porous materials fitting into these lab on PCBs systems. And on that front, we can see 4 different areas where we can see porous systems working. The first one is energy storage. And the reason for that is that the electrochemically synthesized porous materials, especially, can be readily tuned in terms of structure. And thanks to that, we can expect to be able to develop very, very high energy density systems or very fast charging systems. And both of those options are really, really interesting for biosensing devices commercially, especially for wearable devices, such as a continuous glucose monitor, like the ones that people just stick on their arm and have for a couple or 3 days. And the reason for that is that it's very common that one of the most limiting factors on the lifetime of those wearable systems is that the battery runs out quite quickly. The second area is sensors. Thanks to the pore system, it's very interesting to develop highly sensitive sensors by measuring how these pores are getting occluded by the biochemical interaction. Of course, there is another very straightforward approach. They are pores, they can be tuned, therefore they can be used as filters so that we can filter out proteins from viruses, from bacteria, from larger cells. And finally, I'm not going to get into too much of the physics, but porous materials can also be turned into pumps that have no moving parts. And that's an interesting system for all lab-on-a-chip devices, because when you start having movable parts, those are normally the first things that break. So having pumps that have no movable parts and that work with a potential applied, they are very easy to integrate in a disposable system. So that's everything I wanted to present today. Thank you all for your time and the opportunity to present the company here. And if you want to contact us, you can do so through our website, www.matthiassensors.com, or you can also contact us through our registered office at Matthias Sensors Limited in Glasgow. Thank you so much.
Thank you, Gerard. Congratulations on your talk. It was very, very interesting. And thank you for bringing a view on the possibility and the lot of expertise required for the realization of on lab on a chip, and specifically on PCB. If there are any questions, we have some time.
Eleonora, I have a question for Gerard.
Luis, please. Thank you.
Okay, thank you. Gerard, thank you for your presentation and well, your ideas about the future of the materials, porous materials. My question is about the porous materials. Do you think they are, well, every material present different properties and different, you know, characteristics, but what is the material? So what do you think about could be the most promising material applications so for your systems?
Okay, so I'm just going to answer this question regarding the two porous materials that I know most from, which are porous silicon and porous alumina. So let's go topic by topic. So in terms of energy storage, both porous silicon and porous alumina are very interesting, but perhaps for different types of applications. So porous silicon, I think it's very interesting for developing lithium-ion batteries because there is a high theoretical energy density with between silicon and lithium, but there is a practical issue, and that's the volume expansion of flat silicon wafers. So on that front, I think porous silicon can provide a good insight. In terms of porous alumina, I think they might be a little bit better suited for supercapacitors, more specifically pseudo supercapacitors, where you can have a very good engineering of the pore structure, and if you are able to completely use that structure to implement pseudo-capacitive materials, you would be able to have very high energy density and fast charging supercapacitors. In terms of sensors, I would say probably for PCBs, I would be most interested in porous alumina, and that's because you can. There are some types of PCBs that already have aluminium coatings on them. So from my point of view, in this sense, it's easier for me to integrate porous alumina systems. In terms of filters, I would also go to porous alumina because the pores are more narrow and not narrow, more defined and straight. And also the porous alumina has a bit higher stability in water. And in pumps, both of them would work, honestly. I have not tried both of these materials, but I would say both of them should work pretty well.
Thank you, Gerard. Are there any other questions? No? Okay, if this is not the case, then we can close today's session on time. Thank you to the speakers of today. And I will briefly share my screen so that you can see the following event. You should be able to see my full screen. So the next webinar for the Netpore Cost Action Project will be on June 9th. There will be two speakers from the university, so Professor Knežević and Professor Salonen. I hope I got the pronunciation correct. And please refer to the website for the updates regarding the events and the title of the talks. And you can always register in advance so that you do not forget by clicking on this button on the left of the page. And the website is one reported on the top of the slide. So thank you for attending today's session. And you can always look at the YouTube channel to review the webinar. Okay, Then thank you very much for your presence, and thanks again to the speakers of today.
Thank you. Thank you very much. Bye-bye.
Thank you. Bye-bye.
Bye.
Thanks. Bye.
Bye. Thank you.
Thank you. Bye-bye. I'm not sure if the registration will close by itself or if I should do something.
I think you can close the Zoom,
Okay.
and then the session will be closed.
Okay, so the registration will finish. Okay, so thank you. Have a good day.
Thank you,
Bye-bye.
Eleonora,
Bye.
for sharing the session.
Thank
very
you.
much.
Have a good day. Bye-bye.
Have a good day. Bye.